everyone. Thanks for showing up to our 11th edition of our Experiential Science Talk series, where we invite experts with lived experience to share their knowledge and provide new research directions in youth mental health. So there'll be time for questions at the end if we have some time. So today we have the pleasure of welcoming Chantal Murthy. So Chantal is currently a volunteer research assistant at the University of Washington's Spirit Center. She's helped with the Washington State CAPS 2025 campaigns provider database creation. And today she'll be presenting research on Prof. Ari Rappaport's membrane hypothesis of schizophrenia that outlines a compelling root cause conception and treatment for schizophrenia. So without further ado, welcome Chantal. Thank you so much for being here.
Thank you. Yeah, so please interrupt me with any questions if you have any. I want to be as interactive as possible. So yeah, basically I'll give a bit of background. Briefly, some of you may have met me at the April Discourse Conference, but I have been labeled with diagnosis of schizophrenia by a number of clinicians, sometimes saying I don't have, sometimes saying I do. Nevertheless, I have dedicated a ton of time trying to figure out what is schizophrenia, what the current treatments can do for it, where there are treatment gaps, also relying on my own personal experience with the medications and seeing them not always be very effective for me. And so towards this end, I was searching a lot on Google Scholar for like different kind of keyword combinations to see if people had written anything about the connection between schizophrenia and the cell membrane. This was inspired because of some treatment I did for my chronic fatigue syndrome, where I was infused with phosphatidylcholine based on somewhat arcane procedure lost to time that was used a few decades ago, and informed by the late David Horobin, who was an eminent scientist in the field of cell membrane pathology as explanatory in psychiatric diseases, his book. And I was just curious if there had been further work done since Horobin's time on either... Yeah, on bringing to light like either clinical trials or just more research expanding the efficacy potential of Horobin's work. So towards this, I was just Googling like PUFA and schizophrenia and trying to find papers that were written more recently than the early 2000s. And I came across a archive article that was written by a computer scientist of all people, named Ari Rappaport, and I was very perplexed and intrigued by this because it was not what I expected. And I decided to do a careful study of it to see like whether it seemed plausible and whether to see if any more work had been done. So that's kind of like what started my interest in this particular way of thinking about schizophrenia. And I have the link to the archive article here in the description. So yeah, before I get into Ari's PUFA theory, as I was just saying a moment ago, there's been a lot of work a few decades ago or more on trying to understand the contribution of cell membrane differences as explanatory for not just schizophrenia, but a number of mental and neurological conditions. And this has been distilled with the most sort of depth in phospholipid spectrum disorders in psychiatry and neurology. The last edition was published in 2003. There hasn't been a lot of follow-up on this work. I'm not exactly sure why, but I ran into this because of treatment for my chronic fatigue syndrome, and I just wanted to know from the practitioner, like what sort of theory was informing his infusion of me with phosphatidylcholine to help me. And he mentioned this book. So that's kind of where I then started to see mentions of beyond just fatigue, mental conditions as well. So I just have here a few excerpts from this book. I haven't read every page of it in detail, but I've seen enough of it that it definitely has elements that Ari also used, perhaps not necessarily reading this book, but referencing some of the same references that are used in this book. So there's stuff about, which I'll get into, an enzyme called phospholipase 2, and how there's abnormally high activity of a particular variant that is implicated in depleting cell membranes of arachidonic acid and the schizophrenia population, which is here, this bar graph. And I just wanted to show a tidbit of how some of this work has been out there even in the last few decades. And similarly, Patricia Kane, who was the wife of the late Edward Kane, who was a pioneer in kind of developing like healthy like sources of omega-3 and omega-6 for human consumption, and like a really revolutionary scientist in trying to design really bioavailable sources for that. His wife, along with some other scientists, wrote a book called the Detox Book. And again, it also probably builds on Horobin's work, but it also mentions this IPLA2, which is the particular variant which I'll get into in the talk that's implicated in schizophrenia membrane pathology, as like important to consider. And I also have here this test, which is actually still being conducted at the Kennedy Krieger Lab at Johns Hopkins, called the red cell lipid biomarkers test. And basically it gives a very, very detailed breakdown of your cell membrane kind of signature, way beyond just assuming that you're deficient in omega-3 fatty acids, which seems to be the sort of current like messaging, that generally like people's diets are lacking in omega-3 fatty acids, so they need to eat that, which is somewhat reasonable, but the real picture is much more nuanced. And often in schizophrenia, at least subtypes that may have more cognitive deficits, which is also mentioned in Horobin's book, omega-6 may actually be more depleted, particularly arachidonic acid, which will be explained later on the slides. So just taking omega-3s may not be sufficient or even tip the balance in an unfavorable direction. So it would be really cool in the future if there was an intersection of research work that used not just neurophysiological data or cognitive batteries, or even just first-person report, which are all solid, but also maybe looked at cell membrane signature as informing the efficacy and treatment direction for a certain thing given to patients. So that's just my take, but I just wanted to show that. Now I'll get into the actual PUFA theory. So yeah, and I'll just give a brief overview of why this matters. I'm gonna make this a bit, no, I think I'll just make it a bit smaller. Okay. Maybe I'm sorry, I'm just gonna max this out and I can't see. So, okay, sorry. Ari Rappaport, again, I mentioned, was a computer scientist. He unfortunately passed away from cancer a few months ago, but he dedicated the last few years of his life towards really refining his theories and getting them out there. And remarkably, he became a neurologist, kind of psychologist by hobby, and might be suspicious, and he acknowledges this as well, like why his theories are not published in peer-reviewed journals, but it's mostly because he was in kind of a rush to publish his theories before he passed, but his theories have been actually picked up and recognized by the Nobel laureate in chemistry, Roger Kornberg, who described Rappaport's work as bringing exceptional insight and analytical ability to the entirety of brain science. And notably, again, if you can take his word for it, I actually met him before he passed, luckily. It was a very special meeting. I didn't know he was going to pass. I got an impression he was very sincere and authentic in his work. So I would take this at face value, or I did at least, that he read close to 500,000 research articles to develop his theory, suggesting a thorough coverage of the literature. I just have the link here. And again, it's like really interesting because he has kind of a complete theory in his own words for each DSM entity, if you will, or almost everyone. And the biggest thing is not necessarily that his theories are perfect or need to be implemented right away, but that really there's been a sort of lack of creativity in treatment for schizophrenia. It's been quite like much of the same, except for the CAR-XT drug that came out a few years ago. And the medications have serious side effects, which I'm sure you guys all know about. And they often don't treat the root cause of the condition, nor do they adequately address cognition. So whether or not you believe or, Ari's theories, I still think they are an interesting intellectual exercise to think about the condition of schizophrenia from a different angle. So towards this, I'll just introduce two important terms which you guys may or may not be familiar with, which will come up a lot in the theory. One is PUFAs, which are polyunsaturated fatty acids. These are basically fatty acids that are embedded in every cell's membrane. They're vital for structure and function. Unsaturated means that there are at least 2 double bonds in the structure. And the biggest PUFA of interest, which I've also kind of mentioned a second ago, will be arachidonic acid, which is also abbreviated as ARA. And then there's the class of endocannabinoids, which are basically lipophilic molecules that have similar signaling properties to compounds in the cannabis plant. And of course, endocannabinoids have widespread brain activity and help neurons communicate and are related to emotion and cognition. So again, the 4 kind of aims of the PUFA theory of schizophrenia, which is kind of abbreviated by Ari as P*C, is an attempt to unify symptom dimensions into an original mechanistic explanation. And then also to kind of explain the origin and the maintenance of symptoms in chronic psychosis. And again, it could be argued that chronic psychosis that he refers to is classic schizophrenia, though as I will bring up later, I am not exactly sure if his theory could be generalized to explain other types of chronic psychoses. And also explain why long-term use of traditional antipsychotics like D2 blockers may even worsen cognition for some, but using kind of elements in his theory. And also suggests new treatment targets to at least explore, namely to inhibit the collapse of cell membranes due to excessive release of arachidonic acid, a type of PUFA. So I'll get into the first aim, which is the claims on symptom dimensions. And there's a bunch of text here, but I'll just kind of get into the bottom line, which is that basically having excessive release of arachidonic acid from the cell membranes, which itself is mediated by a particular enzyme that I'll get into, causes endocannabinoid dysregulation, and that can explain the sort of disconnection that is present in people who have schizophrenia, and also explain the sort of reduced coordinated brain activity that may be observed. And Ari goes on to kind of explain the positive symptoms and the negative symptoms and the cognitive symptoms as all somewhat explained by this disconnecting switch effect caused by excessive endocannabinoid release through excessive arachidonic acid mediated degradation of the cell membrane. And of course, what causes the condition to sort of develop as well as be maintained he suggests that, at least in the discussion I had with him in some of his videos, the precise array of causes that may lead to this effect is not entirely clear, but there is a chronic stress and or genetic susceptibility that can upregulate the enzyme which strips the cell membrane of arachidonic acid, and this enzyme's name is IPLA2. And as I will show in a diagram later, this sort of stripping by IPLA2 can start a vicious feedback loop, causing discoordinated network activity, which can actually feed back and actually worsen stripping of arachidonic acid from the cell membranes. And then what can maintain the symptoms? Well, it's really kind of this vicious feedback loop, which I will show in a diagram in a few slides. And unfortunately, it is observed that there are attempts to compensate for this discoordinated network activity with actually a reduction in CB1 receptor density in people with schizophrenia, in their brain, that is. But the overwhelming excessive release of PUFAs causing a flood of endocannabinoids ultimately seems to kind of cancel out this compensation. Yeah, so the next slide I'll show you, I don't mean to overwhelm, it's just my attempt when I was reading to try to keep track of all the connections between somewhat novel terms. I would love to hear from you guys how, when you guys study a paper of equivalent kind of terminological complexity, you keep track of all the connections and you kind of make sense of it. And so the other thing I'd like to say is that I did talk to Ari briefly, asking how widespread the particular dependencies in the next diagram I'll show are, in terms of being region specific in the brain or widespread. And he said that the most concentration should be in stress or emotion related areas, but he believed that the kind of feedback loops occur throughout the whole brain. And then also, basically, I sometimes would just draw a connection between an arrow if the paper explicitly said that. So for example, if it said that arachidonic acid interferes with nicotinic acetylcholine receptor function, I would just draw maybe an arrow between these, even if maybe there was like a longer pathway where via endocannabinoids, it could interfere with nicotinic acetylcholine receptors. So there should be more nodes in between ARA and nicotinic acetylcholine. So diagram is just very preliminary. I would love to hear from you guys how to more sensibly understand these kind of papers or diagram your thinking, because I'm still kind of a novice at it. And then finally, basically, actually, I'll just show you the picture, but I just said green arrows are positive flow. So if there's a positive relationship between two entities, and red is kind of inhibitory. And if there's an arrow entering a box from the left, it means that that box is being influenced by this other box. But if it's exiting from the right, it means it's influencing. So again, this is just my attempt. I would love to hear how to do a better job. But yeah, so I tried to make the text here of IPLA2 activity quite big to highlight its central role in explaining kind of the symptom maintenance. But basically, yeah, a stress-related period can, through either genetic or other means, upregulate IPLA2 activity. And that can, of course, as you can see this green arrow, lead to a bunch of effects, including like reduced allergies, as well as increased, increased 2AG synthesis, and basically like you can see that, so this is the important, and of course, most notably, this box, which is increased arachidonic acid release from the membranes. And there is like this feedback loop back from like symptoms to what started them, which is through like agitation. So again, what I want to note in this diagram is that drugs that are on the market for schizophrenia may only like target, D2, SIR2A, and I guess with KarXT, the muscarinic acetylcholine. But like there's many other, neurotransmitter systems, and there's also things upstream of that don't always seem to be prioritized. So I just wanted to point that out. Yeah, and then this is basically like the third point on the claim of PSC for like how antipsychotics can affect symptoms long term. Basically, it's not clear whether like antipsychotics necessarily stabilize the membrane long term away from excessive release of ARA. And there's even some evidence that suggests that some antipsychotics could actually activate genes, and namely this would be the SREB genes that can actually increase IPLA2 and sort of worsen this process. So it's kind of a complicated matter. Do they actually help the root cause or worsen it? It's kind of arguable that they might even worsen it. But most importantly, we know that in general, antipsychotics on the market rarely treat the symptoms beyond the positive symptom dimensions. So regardless of the sort of mixed efficacy on treating the root cause deficit from antipsychotics on the market, clearly more is needed to treat other symptom domains. So yeah, some of the treatment targets that Ari proposes are obviously to reduce IPLA2 activity through IPLA2 inhibitors. Interestingly, there are malaria drugs which also happen to be IPLA2 inhibitors. And there is this interesting correlation between lowered schizophrenia rates in sub-Saharan Africa, where malaria is often present and where anti-malaria drugs are administered. Now, whether that's spurious or not, it does give some interesting credibility to the fact that anti-malarial drugs could potentially reduce this sort of excessive ARA release from membranes. And also, like Ari briefly mentions that there could be like some interesting drug class that could co-stimulate, agonize D2 and CB1 receptor, non-agonized CB1 receptor to somehow rescue plasticity. This isn't very elaborate on, but it is a treatment target he suggests in the paper. And then finally, he mentions NAC to help increase, basically in this diagram, arachidonic acid utilization for prostaglandins instead of anandamide synthesis, because he goes on to say that anandamide, you can see it's through there's increase in anandamide from arachidonic acid. Anandamide can also, he says, dysregulate some of the coordinated network activity. So yeah, let me see. Yeah, and then of course there's a lot of open questions I have from this. So I wasn't super clear when I was reading this how well the theory distinguished mechanistically between positive symptoms and mood symptoms in schizophrenia. Now I know mood symptoms aren't specific to schizophrenia, but it would still be kind of interesting to see like how it could be explained better. If there's sort of like unique cell membrane signature for mood dysregulation of schizophrenia versus mood dysregulation in other conditions. I felt like this section could have used a bit more explaining. And also, I didn't see a lot of depth on distinguishing mechanistically between positive symptoms and disorganized symptoms. Again, whether these are truly independent symptom domains is itself a bit contentious. Some would argue they're not really unique domains and there's more similarity between them than meets the eye. And it's kind of an absurd question to try to propose in the first place. But I would be interesting in the cases of explaining like purely hebephrenic or like disorganized subtypes, whether they would also follow this sort of pattern described in the P*SC. And also, of course, stress sort of independent causes of chronic psychosis. And whether these are fully stress independent, that's arguable, but generally not something that just sort of arises organically or genetically linked, is seeming to be explained by, things that are arising without a sort of infectious etiology or genetic link seem to be explained by P*SC, but what about things that are? So like for example, the 22X deletion syndrome, or like things where there's an autoimmune condition that can result in chronic psychoses that mimic like a schizophrenia diagnosis, or like Lyme brain. And also like frontotemporal dementia that can result in like maybe a different sort of profile of schizophrenia, but might also have some resemblance of some symptoms. So I'm curious if the syndrome described really does match classic Bloy-Laren schizophrenia. When I spoke to Ari, that's what he seemed to believe, but again, it remains to be proven in the real world if that's true. And then also finally, there's like this sort of trope about sort of neurotoxicity in many cases in schizophrenia, just from like the way that the neurotransmitters and cells are communicating to each other. But there's a lot of debate on that and whether like that really is reversible or even true in many cases. And so one thing is there's sort of like this glutamate excitotoxicity sort of model, which talks about excessive cell death in the first few years of many people's presentation with kind of first episode psychosis. Now, is that really sort of permanent sort of neurotoxicity observed, or is it like more related to disuse, or is there like actually no connection between the glutamate toxicity seemingly and the neurotoxicity, and there's a separate mechanism? I feel like these questions are not always super well explained in the literature, and so I kind of wonder like how well the P*SC could explain it. When I spoke to Ari, he seemed to believe that the sort of narrative about excessive cell death was somewhat of a myth in schizophrenia. But again, I would love to hear your guys' thoughts on that. And then also, of course, you guys might have heard about Alzheimer's and how like there's been a lot of controversy among the years in like the way that some results have been fudged in very early publications in the 90s. To sort of bolster the credibility of the amyloid hypothesis and sort of treatment target model to eradicate Alzheimer's, and how there's like this renewed focus on maybe like looking at the cell membrane. So one of the components that's been of interest is plasmalogens. This was popularized by Dr. Dion Goodenow in a lot of his work, and it has like some explanatory power for some of the kind of cholinergic signaling deficits observed in Alzheimer's. Now, one thing is sometimes it's observed that there's sort of neural inefficiency either through EEG or just subjective reports in people who might experience schizophrenia. And I wonder like how well could integrating plasmalogens into the P star ST theory bolster it, and like how does it all relate to the sort of central deficit that Ari kept talking about with excessive IPLA2 and arachidonic acid release. And then, of course, I already kind of mentioned this point about the regional specificity of all the feedback loops and whether region-specific inhibitors need consideration. I'm not super sure on this point. I seem to not think so, but again, it really remains to be seen in trials and like the kind of real world. And then finally, like a general question about how to develop kind of mechanistic models that are maybe not computationally based, but just based on like scouring tons of literature. How to determine like what is the appropriate strength of connecting two concepts. The model drew on a wide range of studies, from cell cultures to human research to back up each link. But in general, like if you saw a diagram like this or you were trying to put it together, like what would be your approach to like have the right strength of connection between two concepts so that it like tipped the like favor of like explanatory power in the right direction and didn't privilege all the nodes as like equally important? This is something really difficult for me to figure out. I'm not sure of the right approach, so I would love to hear from you guys on that. Yes, and then finally, comorbidities. I didn't see a lot explaining like how the theory could explain comorbidities of conditions. What's interesting is he has sort of like a complete theory in his words for each or many of the DSM entities, both psychiatric and neurological. But I wonder, like, for example, there are people who have like both OCD and like schizophrenia, schizoobsessive disorder. It's coming more to light. Would you then sort of stack the treatments for those two conditions, or is it a totally different like condition itself? So that's something I'm still not clear on. And then also, he already didn't take a symptom specific approach to develop his theory. As I pointed out close to the beginning, he did conclude that his theory could explain positive, negative, and cognitive domain of symptoms, but he didn't start off saying, I want to explain, for example, some like neurophysiological abnormality, or I want to explain like why people might exhibit disorganized speech, or I want to understand why people hear voices, and then proceed from there. It seems like he more like just assumed that the word schizophrenia had a lot of variability indexed in it, and then from that did a thorough search of the literature to then find patterns. So again, that's like a slightly different approach. So it's kind of like, how does this approach compare to researchers who take increasingly granular focus onto different symptom domains to find differing mechanisms, and which one might lead to more accurate results and better treatments? Kind of a difficult question. I don't know the answer, but I just thought I'd throw it out there. And then finally, kind of more of like a really like how to collaborate between diverse audiences type question. There seems to be a sort of polarization between the camps of people who need to precisely define what is pathology and study and treat it, and also the camp, and then on the opposite side, the camp of people who want to increase societal acceptance, not just in the sense of this person has an illness and we need to respect that, but more normalize and say it's sort of a variation on human difference of like condition, some of which you might even include schizophrenia. Now, these two camps exist, but in order to each maximally pursue their agendas, must they be siloed, or is there a way for them to actually work together? Or is that an impossibility based on the kind of aims they're pursuing? Because it's not kind of like if there was a sort of treatment in theory for schizophrenia that could treat it quite early, then would that sort of decrease the need for societal acceptance or deprioritize research there? Or would that still matter? And the people who don't want to necessarily change who they are would still want to be accepted, so that would matter. Like all these kind of things, I don't know. It seems like kind of difficult to figure out. And then finally, we hear a lot about cannabis and schizophrenia and how it can sort of worsen or even bring out a potential illness of schizophrenia or condition, I guess, of schizophrenia. But what about caffeine? Now I've seen a lot of mixed results, and also in my own like kind of experience with caffeine, felt really conflicted about this one. We hear about oversensitization of CB1 receptors, but by cannabis. But what about from caffeine? Now there is some literature that supports that. However, some people say that caffeine can actually help them focus and help them concentrate in a way they wouldn't be able to without it. But other people, like myself, when we use caffeine, it may even like bring out previously latent mental noise, if not actual psychosis. Though I'd say it's kind of a spectrum. And I wonder if that's related to sort of like the CB1 receptor sensitization affecting the striatal dopamine system. And maybe like people who have different types of symptom profiles might be experiencing caffeine very differently in that regard. And I bring this up because of the connection to like endocannabinoid dysregulation as the thing that affects the sort of network disconnection in the P*SC model. And I wonder if caffeine could also kind of not independently of the membrane degradation sort of cause that endocannabinoid dysregulation. And I think probably, yeah. And then finally, we hear a lot also about CBD, cannabidiol, as like kind of counteracting some of the symptoms of schizophrenia or whatever. But like one of the things is that CBD can increase TRP1 activation, TRPV1 activation, and that may worsen hyperexcitability of the glutamate system, cause disinhibition. And purportedly, if glutamate excitability is the thing, or one of the things driving neurotoxicity, like what could be done then for the people who might exhibit that sort of effect from CBD? Now, I say this mostly because of my reaction to CBD, so it's a little bit personalized, but also things I've heard in people who might have stronger mood symptoms, but also some like neural noise that might, for lack of better words, be considered psychosis. And I also kind of mention here, again, this is all sort of special case to try to understand my own reaction to things. If the CB1 receptor downregulation that I mentioned close to the beginning of this talk as a compensation to reduce the endocannabinoid flooding could itself actually be sort of like the reason, at least why, in my experience, when I take CBD, I go into a sort of drug withdrawal kind of state with cold sweat, shakes, and sensory hypersensitivity. I don't really have a lot of evidence for this, but I have unusually heard of people who have these sort of reactions to CBD, and I wonder what's going on. And then finally, like how much of network disconnection, kind of as we observe in this PCRSC model, and also just in general, like in patients, has treatment target overlaps with chronic fatigue syndrome? And I say this because during moments where I felt especially fatigued, I feel like I've exhibited more kind of negative formal thought disorder in my in my head at least, even if I might cover it up in my speech somewhat. And I wonder like if there's some kind of overlap between those two entities and like what could be done. And then finally, there's sort of this kind of narrative in the literature that talks about schizoaffective, or like basically the presence of mood symptoms along with schizophrenia symptoms as indicating a better prognosis in terms of cognition, functionality, than just the schizophrenia symptoms alone. I wonder like what is going on there, because moodiness could seem to also worsen stress, which could then worsen cortisol mediated degeneration of membranes, which, if you recall, was what sort of begat the whole thing. But, or if it just means that some of the feedback loops between all these guys are a little bit less coupled, and people who might have the moodiness as well, and so separate. I'm not sure, but it would be really interesting to explore that. Yeah, and yeah, that's all. So yeah, and just like, what would you like to know more on, and just any kind of feedback. Thanks. Hushman?
Yeah, I have a question. Thank you
Yeah.
so much, Chantal, for
Yeah, no problem.
I found it very insightful.
Thank you.
And my question is to do with, phosphatidylcholine,
Yeah.
and a lot of things that you did discuss are implicated in the inflammatory pathways.
Yes.
So you mentioned you received an infusion of phosphatidylcholine, am I right?
That's correct. Yeah.
Was it intravenous?
Yes.
Would you be open to sharing? What was your sort of response to that?
Yeah, I can.
Did it sort of ameliorate your symptoms, other symptoms, that sort of thing?
So I struggle with chronic fatigue and just really, really deep exhaustion and unable to sort of sit up and talk. And so this was after a period of particularly bad fatigue where I was bedridden most of the day. I couldn't think very much. And basically, I found a naturopath who seemed to do this unconventional approach, and he talked about like, well, first what he did was he actually did a red blood cell fatty acid analysis on me. Now that seemed more just like as an index because it didn't really change the treatment he followed, which was actually based quite carefully off of the detox book, which I have a PDF of, but it's no longer in print because of, I can explain in a bit, some lawsuits that were filed against Dr. Kane because of just this one ALS sort of amyloid lateral sclerosis kind of patient mess up where patient thought they would get better and she said they might not, and there was some, but to help people effects I experienced were actually like this profound sense of mental calming and clarity, but not in like a drugged sort of sedated way. Now at the same time as when he was doing these infusions, he also basically was providing me some two different oils, one of which was like this phospholipid complex. And then it had like beyond just phosphatidylcholine, had phosphatidylinositol and a few others. But and then the other one was like this very like kind of a proprietary blend of different fatty acids. Now, the particular compounds or the particular supplements he gave me were actually like they're only produced by his mentor's company, but the closest analog he says that are on the market are by this company called Body Bio, which actually is Patricia and Edward Kane's kind of descendants' company now, and they have like these sort of micellized forms of phospholipids. I actually have used these as well once I ran out of the one that my naturopath gave me, and they do work somewhat equivalently. But yeah, once I basically did those infusions and started to use those supplements semi-regularly, because I was having very strong mood reactions to these things, which was unexpected, I started to feel profound change in like my ability to like think abstractly and like not have a lot of mental noise. Like my auditoriness of my own thoughts was lower. I felt like I could just implicitly think and like in a way that I seemed to struggle with before that. It almost felt kind of like doing transcranial magnetic stimulation, but like the effects seemed to kind of stack. And when I noticed my brain kind of going back into more neural noise or like kind of getting into those more deep fatigue states, I will still occasionally take a bit of that body bio and try to get back up. But again, that's just my very personal experience with it. I haven't seen a lot of people talking about it, but it seems like something to explore at least.
So these are pills you're taking, yes? Supplements?
Yeah,
pills.
the one from Body Bio, they're pills. Yeah, there's also liquid form.
Great.
Great. Thank you, Chantal. Can I ask a question? This is very nice. Thanks for explaining patiently this, long-standing notion. I didn't realize that Ari Rappaport's work covered all of this, and I didn't realize he passed away. He's one of those people who have written a lot about many things, I think, many psychiatric disorders. Anyway, so coming back to this PUFA point, this membrane, lipids in the membrane, cell membrane problem, I would like to know what you think of. You didn't mention omega-3 will not work, but there was an initial trial a long time ago in Australia done by somebody called Aminger. It was quite an interesting trial. They ended up showing that people at risk of psychosis, not people with schizophrenia, but people at risk of psychosis, when they take these higher doses of PUFA for a long time, omega-3 fatty acids for a long time, around 6 months or so, I think, I don't remember exactly the time, but they showed that around 1 out of every 3% or 4% who took this prevented onset of psychosis. So the number needed to prevent or number needed to treat, so to speak, N and T, was pretty impressive, something like 4. But it wasn't replicated. Somebody else tried to do the same. I think the same group might have tried again. But anyway, the story became weaker when there was no randomized controlled trial evidence for correcting any of the lipid abnormalities. So one thought that some of us think is the idea itself is not wrong, but application wasn't correct because we don't know which is the group of people who have this abnormalities. There's no way to select a subgroup of psychosis folks. What do you think about that?
I think that's super relevant. So I just wanted to quickly share something, which I think will give some more clarity. So basically, I have the, I don't know why it's up. I have the first edition of the Phospholipid Spectrum Disorder in Psychiatry, and it gets into a ton of detail about kind of like different ways of treatment. And I think it has a chapter about omega-3, new strategies for treatment. I think just like you said, it's not that necessarily omega-3 is not helpful. It's more like we don't know which subgroup of people would be like the most responsive to that. And also like without like a clear sort of lipid profile, which I actually got one done, with the provider. This is not proprietary, but I'll just share it. So like it's hard to say like which thing you want more of. Like some people, they might not actually need omega-3 and they may actually be very deficient omega-6. Like so it's, I think, spectrum of like fatty acids and but of like what someone's membranes are looking like. And again, you might say, how is this coming from the brain? There's some research that shows a correlation between peripheral red blood cell membrane composition and the brain. So that's kind of, but yeah, there's I haven't read every bit of this book, but it does have a whole chapter on omega-3 and schizophrenia,
Yeah.
but also on many other,
Mm.
sorry, it's in the table of contents. I can email this to you if you'd like.
Yeah, we have. I mean, it's really an interesting idea. Alvin just put another RCT to show that the second result was negative. There are many negative RCTs on this omega-3 now. I put a link on the original
Okay.
study. It's a short
I
report.
think,
Read
yeah,
it.
it may just not be sufficient to address like
Yeah.
these upstream sort of degradative processes. So like, yeah, it's kind of.
But if if we find a way to treat this way, these tablets or these medications are much more acceptable. The biggest problem people have with omega-3 is after you take high dose, you get a fish burp. It's quite smelly. That's the only problem.
Yeah,
You
there's
don't get
nothing
yeah.
compared to some of these other
Yeah, and
effects.
so
Yeah.
so placebo control is very hard for this. This is actually a valid scientific point as well. Placebo control for omega-3 fatty acids wasn't easy at the time. Some suspicion was people who took it actually knew they were on it, so they were
Oh,
reporting improvements. I think it might have been better now. I haven't looked at recent literature how they make a placebo that smells like fish. I don't think it's impossible, but this is
yeah.
yeah.
Well, I would be curious on that point, like how phospholipid supplementation helps people with
Yeah.
schizophrenia. I think there's been just a handful of trials and stuff, but not much. So.
Mm-hmm.
Okay, we have 5 more minutes. Oh, I'm just saying, go ahead. Anyone can ask a question.
So the phospholipid trial, there's one on clinicaltrials.gov. You can probably pull it up, but it's been, yeah, there's one in 2020 that's been paused.
Okay. I'll find it, but
Yeah, I can send it to you. I can send
sure.
it to you later.
Okay. Yeah. Again, my experience was really unusual. Like, I don't know if other people react that way. I had predominantly kind of thought disorder symptoms, not like the hallucinations and delusions or whatever. But it really helped my neural efficiency in a way that nothing else has, other than TMS. And I'm not sure what's going on there, other than some kind of white matter integrity boosting something. But like, yeah.
I know you had so many open questions, Chantal. If there was any other question you wanted to ask before we ended, of course this will be recorded, so people can always come back and look at it later, and we can continue the conversation that way. But if you have any closing points.
I would love to keep talking about this sort of thing and just like understand, especially the point here. I think this one is general enough. I'm really lost on it, like about kind of this wiring set of aims between trying to normalize, certain mental conditions, and then also really precisely almost trying to extinguish them, not necessarily in a sort of negative intent, but like, I just don't I don't see a lot of crosstalk between these camps. I really don't. And I'm wondering, is that just inevitable, or is there a way to fix that? So.
Yeah, I'm sure others have more opinions on this. Irnes might have a strong view on it. I'll just add 2 pence to this. I think there is this phenomena called clinician's illusion. And I'm biased, of course, I'm a clinician. And clinician's illusion actually means we get to see the world through the lens that's given to us. Like all of us do that. And clinicians end up seeing the really sick, the really unwell people, especially in psychiatric hospitals, when you work in a psychiatric inpatient unit or a psychosis clinic. All that you see is you think of this as like a spectrum. You only see this far end of this Gaussian curve, which is very severely affected people. So you create this impression in your mind that this is an illness that's kind of it is severe and every symptom has to be extinguished, every symptom has to be addressed in order for people to get better. Now, that is biased, not wrong, biased. I say not wrong biased because it's not wrong in the sense that they're not, people who are only exposed to these kind of severe end of the spectrum are not wrong when they say you need to get better in terms of symptoms for you to go back and integrate with the society and do things. But it is wrong in the sense that they're making that conclusion based only on that spectrum that they captured, which is the severe end of the illnesses. So this illusion is, this is a constant, it's a philosophical problem as well, because when you speak to a person who's recovered, who's doing well, they will just not agree with this perspective that you got to treat a symptom in order to get, functioning better. It is shifting with, programs like early intervention now. We see people getting better. So I think there is an exposure of different spectrum of this illness, but I don't think this is the only answer to this issue. The philosophical differences exist between different groups of people, all with a good common goal. We want to eradicate and extinguish this problem, but we all take a different part of the animal, and one of us call elephant, the other person calls rabbit. It's a big challenge.
Yeah, totally.
Yeah, and I really appreciate what you said there, Lena. Like, there's definitely like multiple truths with psychosis, right? And I'll be honest, like some of the scientific language stuff, it just went over my head. I'm not gonna pretend like I knew what you guys were talking about when it got very that scientific. But what I really appreciated was the piece around Chantal is trying to look at, what are some of these long-term impacts of the medications that are presently prescribed? Because, it feels like we all feel it around us, and at every clinic we notice the long-term effects, but nothing is really done to really look into these impacts that it has on people. So I really appreciate that we're at least searching for another truth as well. And I think the other piece was just around chronic stress. Like in our recent family workshop, I would say every single lived experience speaker talked about stress as a major factor. It was heard and clearly and loudly by everyone in the room, right? But it was frustrating to see how quickly the role of stress was just underemphasized or brushed aside once we switch to medical explanations. And again, it's not that one theory is wrong or right. There's simply multiple truths. But I do wonder if we sometimes favor the one dominant perspective. I just want to say thank you, Chantal, for looking at some of these theories and creating a less of a gap between theory and practice. Like I see a lot of theorizing, but when it's put to practice, I feel like we still get the same effects.
Right.
I'm not sure that people are getting better.
Yeah, totally. There's a lot of detailed literature on different like white matter tracts abnormalities or like functional MRI studies showing this or that, but what does it do? How does it translate into actual interventions? I personally don't see a lot, so it's very encouraging to think there are pharmacological targets beyond what we consider right now in practice that we could explore. So
Tasha, I know we're running late, but is there time for just one quick comment?
Yeah,
of course, please, please go ahead.
The point that you made, Chantal, and Ernest, your response is very important as well. And I think there is a historical bias here, how the field started and what we're doing right now. One is the way that early intervention and youth mental health was conceived. When people realized that you got to intervene early, which is important, which is meaningful, we all accept that. I think it's kind of a no-brainer to say don't intervene early for any problem. So I think we all accept that, early intervention is good. But when it started evolving, it was not ready for prime time, because what people ended up doing was, from this long-term schizophrenia treatment programs like asylums, where we used to use high dose of antipsychotics, we just took those treatments and transposed them in small doses to younger people. We were not really focused on developing treatments that are stress-related, sensitivity reduction, things like that. Even pharmacologically, psychotherapy, pharmacology, everything was taken from this chronic samples, chronic people.
Right.
I use the term in a very respective way. but that was taken and transposed. And this transposition does not work because it's not purpose-built. A lot of people stop taking these medications. In asylums, for example, long-term hospitals in the past, this was never a problem. Compliance was not an issue. People were like, cognitively impaired, catatonic and all, but they were given these medications, and some of them will routinely come and ask for it. So the expectation was that you just give it to young people, they'll take it and they'll get better. It just didn't happen. The recovery rates for schizophrenia, some very hardcore early intervention clinicians will disagree with me because they also have a clinician illusion. They think that early intervention is solving all the problems, a different kind of illusion. But the reality I see is we haven't reduced the frequency of schizophrenia, we haven't reduced the prevalence of it, we haven't reduced the incidence of it, and people who develop this long-term persisting psychotic disorders are still jobless. So we haven't really done much with this early intervention program. We get a different lens, and we're all happy about it when we work in PEP clinics, but the reality still is biting us, and that's the truth, unfortunately.
Totally. That makes a lot of sense. And I like what you said about how transpose treatments from like the 50s or 60s to these younger populations with maybe less severe symptoms without any sort of novelty or like exploration of what really can be done. And so absolutely, there's a lot of that.
Yeah, thank you for that explanation, Lana. I really appreciate that history and transition. And to be honest, I feel like sometimes too, right, we kind of confuse treatment with comfort. Sometimes I ask some people, how's this client doing so and so? Oh, they are doing amazing. Okay. Are they working? Are they in school? Are they getting out and about? Are they chasing girls? Are they being social? None of that. So then I ask, what is so great? And sometimes I wonder, like, how is quality of life defined? Is it purely by clinicians, or should we be more asking clients more, right? Sometimes I feel like when we just ran the family workshop, if we actually let clients just write their own story, what led to their psychosis? At which point did they gain insight or maybe didn't? And what helped them maintain? If we can kind of add to that story or inquire more, maybe we would provide more unique treatment.
Definitely. Yeah.
Okay. I guess we can wrap it up here. Thank you guys for your comments at the end, and thank you so much, Chantal, for coming with this great presentation. I loved how you had all these questions, these open questions for us to think about. So thanks so much, and yeah, I'll wrap it up here.
Thank you, Chantal.
Thank you.
Thank you, Chantal.
Okay.
Everyone, see you.
All right.
Bye. Thank you.